Right, so today we're recording mass spectrometry general sample recommendations. This is Emma Dowd. I'm the assistant director at the IU School of Medicine Proteomics Core. So first off, you want to get your sample into the mass spec at some point. So you need to solubilize and prevent degradation of both the protein of interest and any potential post-translational modifications. So I do want to emphasize that you can go ahead and submit cell pellets and tissue directly to the core for lysis, and we can take care of that sample preparation for you. Our standard lysis includes 8 molar urea, which denatures almost all proteins very quickly and prevents a lot of the degradation that could happen. Most projects, we do use some sort of mechanical disruption to completely homogenize tissue or cell pellets. We use processes such as bead beating, sonication in a bioeruptor with cold water bath, or cryolysis, which is lysing under liquid nitrogen, which is great for samples such as tumors or yeast cell pellets. For label post-translational modifications, it may be a smart idea to include inhibitors at some point when you're either pelleting your cells or lysing your cells or tissue to prevent the loss of those post-translational modifications. So for example, phosphatase inhibitors if you're interested in specifically phosphorylations. One thing I really want to emphasize in this lecture is that detergents may be your friend in most biological applications, but in terms of mass spectrometry, they are your enemy. If you use any detergent anywhere in your sample preparation prior to giving us the sample, it is imperative that you let the mass spectrometry core know that there are detergents present. Unfortunately, almost all detergents are not compatible with downstream mass spectrometry. And we have here detergent contamination of a mass spectrometer is very costly. It can take us a long time to clean up columns, tubing. And on the bottom right of the screen is an example of what you see. So most detergents ionize extremely well. So all you will see in the mass spec is detergent. You will not see any of your peptides of interest, and there's no way of removing that inline or online. So anytime you give us a sample that has SDS, Triton, Tween, all you will see will be peaks of detergent, and you will just get not very good data. So it's really, really important to think about that and have some of your last sample preparation steps, if you're doing your own sample prep, be numerous, copious washes of something with a buffer that has no detergent in it at all. I wanted to show you this screen so you can register as a user at iLab Solutions. This is the new sample submission website that all of the cores at IU School of Medicine are going to start using. And here you can see some of the options for submitting samples through the proteomics core. And you can see you can start with a consult with the core staff, and I always recommend you do that. If you have any questions at all, feel free to reach out to us, and we're more than happy to discuss prior to carrying out any experiments. It's really always easier to think about this before than to deal with, for example, getting rid of detergent after you think you have a finalized process in place. So the most common sample preparation or samples that we have submitted are affinity purification, complex protein identification, or in-gel digestion, recombinant protein analysis, in which case you're just trying to identify a single protein. So for affinity purification applications, this is when you have a tag of your protein or an antibody to your protein, and you're pulling down this protein of interest, and you're going to submit beads, resin, or something that has been eluted from the beads or resin. So again, it's very, very important that you think about how your starting material for this affinity purification is fully dependent on the expression level of the target protein in the system and how well your capture method works. So if you are using, if something is in your sample but at very low abundance, you may have to start with a lot of sample prior to your pull down. Even though mass spec is a very, very sensitive method, you need to pull down some of it. Another thing that a lot of people don't think about is that your antibody or tag may not be very specific. So this can be less of a problem in a Western blot where you can kind of ignore these secondary bands, but we're not ignoring any bands when it comes to mass spectrometry. So your antibody might be interacting with a number of different proteins. And this is becoming a more common and recognized issue with commercial antibodies, but it's something to think about. And that's why we always recommend a couple of rounds of optimization and double checking that your antibody and tag work for your system. Washing of your pull down is also very important. We definitely consider a large volume of washes, at least 10x your bead volume, at least 4 rounds of washing to remove nonspecific binding if you want to ID something, a specific protein. So this higher volume removes all the detergent. High salt can remove nonspecific binding. So this binding might be to some other portion of the antibody or to the beads themselves. You have to think about how there's matrix effects and interactions that happen there. And you're going to pull down all of that. And so the more washes you do, the better. A bait effect when a protein is overexpressed in your system. So if you're looking at interactions of a specific protein and you're overexpressing the protein that you want to see what interacts with it, you might just see that protein because you've produced so much of it that when you submit a sample to us and we digest it, you basically just get really abundant peptides from the bait protein. So you might need to express at a lower level if you're looking at interacting proteins in an affinity immunoprecipitation. And we do recommend that you run at least biological duplicates if you want to publish novel findings. That allows us to run statistics such as Fisher's t-tests. And if you run in triplicate, we can run ANOVA tests. So that's really important to think about just for reproducibility, being really confident of your results. So it's, this is the most common type of sample that's submitted, but some of this background, you may read in the literature that you do a simple immunoprecipitation and you just follow the directions in the antibody protocol. But there's a lot of moving parts and optimization is really important for you to get good data from the LCMS.
Can I make a comment, Emma?
Absolutely.
Yeah, so the other thing, this is Amber Mosley, and so the other thing I would like to mention here is that if you use too much antibody, we'll have actually almost a similar problem as that we have with a detergent. And so if you add an excess of antibody, what ends up happening is the only thing that we'll see in the mass spectrometer are peptides from the antibody. So it's important to recall that antibodies are also proteins. And so when we digest, we'll also have them in the samples in a lot of ways. There are some workarounds around that, but that really comes in with the experimental design. And so if you're concerned that you're going to have too much antibody, there are some ways to work around that, but that's something that you can definitely discuss with us as well. And there are a number of different options that we could do to try to avoid that type of contamination as well. One of the ones that I recommend most frequently is that you do a titration. And so that you know the minimum amount of antibody that you need to use to pull down your protein of interest completely from lysate. And so by doing that, you're able to really reduce the amount of antibody that you add. It also helps to give you some idea of the affinity of the antibody in a very rough term. And so then that can be really helpful when you're designing your experiments. So I just want to make that comment.
Yeah, and I think that goes with this evaluation of antibody efficacy. We see people submit things from IPs or pull downs where their antigen might be there, but maybe it's not the most abundant or maybe not in the top 3 most abundant proteins. And that's when you really need to think about kind of optimizing things where it seems like something might be quote unquote good enough. It might be worth it if you're going to do a lot of long-term studies where you're submitting more samples to optimize at the early stage so that it doesn't complicate your findings down the road.
I will make one more comment on this, that if people come to me and they ask me like a general starting amount, so I do recommend that you start with somewhere around 10 milligrams of starting material for most proteins, which is the larger amount of material that we actually request for some of the experiments that we'll talk about in a little bit as well. So 10 milligrams is what I recommend. And then normally the starting amount of antibody that you want to use is around 1 microgram. And so you'll see that there's a huge difference in the amount of starting material versus the amount of antibody. And so then that's the kind of ratio that you might want to keep in mind if you're wanting to do a starting experiment to be able to do those titrations. So around 10 milligrams to 1 microgram of antibody. So just that last comment.
Great. So another set of experiments that you'll have heard mentioned in previous lectures include the global protein quantification, phosphorylation quantification, or other PTM quant. So for global quantification, I think there are a couple of other lectures getting into more detail on how TMT, tandem mass tag labeling works. So I'll try to focus here on the actual sample submission process.
Yeah, so it'll specifically be discussed in more depth in the quantitation module.
Yeah, so that's in a couple of weeks, I think. So here we really recommend that you directly submit a tissue cellular protein sample to the proteomics core so that we can carry all of these steps out in situ in our lab. We can always discuss if you want to do more or less of the sample prep in your lab, but cells do need to be carefully washed before they're submitted. Most tissues you're going to want to snap freeze so we can do cryolysis of some sort. You're going to need to submit a minimum of biological triplicate for us to do statistical analyses. And if you're running a complex system, maybe you have something from mice where you need to look at sex and line differences. You may, and patient samples, human samples, you may want to run 4 or 5 replicates just to make sure the findings you have from the statistical analyses of the sets of samples are as accurate as possible. So the reason we ask you to submit these samples is that it allows us to prepare the protein, and then we can look at the amount of protein from each sample. And it's really important in this sort of quantification method that we begin the labeling process with the same amount of protein from each sample. There needs to be some way of normalizing across your sets of samples. And we request at least 30 to 50 micrograms of protein, which obviously in cells is not a big deal at all. So for global quantification, we can work with almost any system. You can usually get that much. And the nice thing about this is that for multiplexing, you're actually adding all this protein together after labeling. So sorry for the low resolution image, but essentially each sample will be prepared individually and labeled with a tandem mass tag TMT label, which essentially barcodes each sample. And then when it's mixed together, the peptides are still barcoded, but in the LC-MS analysis, the barcode is the same intact molecular weight for each peptide sample in the MS1 precursor scan. But when it's fragmented to MS2, we use these reporter ions to look at the differential abundance of a specific peptide in each sample. So in summary, for global quant, you don't need as much protein, but we do prefer that you have a talk with us, make sure your cells are carefully washed before submission. For post-translational modification quantification and phosphorylation in quantification is our most common PTM, you need to submit a significant more amount of sample. And this is what Dr. Mosley talked about a couple of slides ago, where because you're doing an enrichment, so any PTM exists at a relatively low abundance compared to all unmodified peptides in a sample. So you can see in this schematic, you have all of your tryptic peptides from cell lysate or tissue, and only 1%, 1 to 3%, or less than that in situations such as ubiquitination, have any of the modifications of interest. So this is why, while running a global sample, you might not see any of your post-translational modification of interest. The other peptides are just significantly more abundant. And so to quantify and identify these PTMs, you need to do some sort of enrichment. For phosphopeptide, it's actually a chemical enrichment most of the time. And we use a titanium dioxide that binds the phospho group on peptides that have phosphorylation. But we still need to start with at least 2 to 3 mg of protein for the serine threonine phosphorylation. So that's for a chemical sort of enrichment of the PTM. But if you're doing an antibody enrichment, which if you think about it is essentially an IP, you need to start with these 5 to 10 milligrams. So for tyrosine phosphorylation, acetylation, and ubiquitination, the way we do the PTM enrichment is actually using antibody on a resin. And so we need this really large amount of starting material so that we can identify the most possible sites for you after enrichment. So it's definitely a different set of considerations than for a global experiment. And you often will want to be running global quantification at the same time in parallel, because it makes sense that if you're interested in a PTM, a specific PTM, you want to know whether the actual protein is changing in abundance or just the PTM. And so it's usually not a big deal because you're starting with so much sample that it's quite easy to, in parallel, do the global quantification. So there's significant more sample prep involved in post-translational modification, and there are definitely some more aspects of the enrichment steps that can be tricky if you need to. So always feel free to reach out and chat with us about it. And then we have a whole section here. There's so many special cases. We have a thermal proteome profiling, different data analysis, publication support. We can pull data from public exchanges and help you do metadata analyses. Proteomics is an amazing tool, and I think that you should definitely consider it in your biological applications. So don't be afraid to reach out. Dr. Mosley, do you have any other comments?
I think that's great, Emma. Thank you.
Thank you.